Hi, good afternoon, everyone, and welcome. This is Pat Furlong for Parent Project Muscular Dystrophy, and we're really thankful and thrilled to be able to bring you today's webinar. Chris Garabedian and Dr. Ed Kay from Sarepta will be talking about eteplirsen. As we all know, we're excited and encouraged about the results that we've seen in both from the World Muscle Society meeting and today we're going to have a review of the data and then answer some of the questions regarding next steps, production, and what we are to see in the future. So with that, I'm going to turn the webinar over to Sharon Hesterly, who will begin to introduce Dr. Kay and Chris Garabedian and start the webinar.
Okay, thanks, Pat. So as she mentioned, we'll hear first from Chris Garabedian, who is the CEO of Sarepta, followed by Dr. Ed Kay, the Chief Medical Officer. And all I'm really going to say right now is just sort of explain the ground rules of this presentation. For those of you who maybe haven't attended a PPMD webinar before, the voice is one way. It's all muted, so you can hear, but you can't ask questions verbally. What you can do is you can ask questions in the chat box with the understanding that we will be answering questions at the end of the presentation, but we're actually going to just be addressing questions that we received in advance. We put out a call for questions, and we received quite a few in advance. So we have a very detailed lineup of questions that we'll go over at the end of this presentation. I mentioned the chat box. You'll still be able to write questions. Please don't hesitate to do that. You can do that. We'll collect those. They won't be lost. If there is anything that is not covered sufficiently at the end of this presentation that we're able to answer, we'll see if we can answer it in by some mechanism following the presentation. So just so everyone is clear, we're not going to be answering impromptu questions during this presentation. And so, that being said, I will now turn it over to Chris.
Sharon, thank you very much. And first I want to say thanks to PPMD. This is the third webinar that we've done facilitated by PPMD, where we have an opportunity to talk directly to the DMD community, and it's very important for us. I will say, however, that we share new information through press releases, through investment conferences, through other conference calls, and so as we've done with previous PPMD webinars, we'll be limiting this to previously disclosed information. However, we know that the community has different questions, different clarifications about data that we have presented, and Ed and I will attempt to answer all the questions that we received, from the community, and we will have an ongoing dialogue. So we really appreciate this opportunity. And please understand, if we can't provide new information, it's because we use our, disclosable event. And we have an opportunity in a few weeks' time in early November. Soon we'll be trying to calendar a date where we will provide our earnings update. And usually in these earnings updates, this will be our third quarter earnings release that will be communicated sometime in early November. That's usually the opportunity where we provide more specific information of our intentions and our plans moving forward. But what I wanted to do today was provide you a brief update of some of the activities that we've had recently. Obviously, we were all very excited releasing our 48-week data on October 3rd, and we proceeded to provide a further review of that data in presentation form at World Muscle Society meeting. That was in a press release. We outlined some of the new information we'd be sharing on October 12th, and we posted the presentation slides that were delivered by Dr. Jerry Mendel at World Muscle Society that took place in Perth on our website. And so those are accessible. Ed Kay will be going over those slides and those data and describe the data that was presented. World Muscle Society was a very important meeting for the company. We were a co-executive sponsor along with Genzyme, and that provided many opportunities to establish a strong presence among the world's foremost experts and leaders in the neuromuscular area, and specifically the Duchenne muscular dystrophy area. We had a lot of informal meetings. We had a lot of one-on-one meetings about how to continue to develop not only eteplirsen but our broader DMD program. We had group meetings with opinion leaders in the space, we had a industry symposium that we put on Thursday night of the meeting that was attended by about 100 to 200 people to hear about the state of the art of Duchenne muscular dystrophy in terms of not only biochemical assays and what are the best endpoints to look at on dystrophin, but also a presentation of kind of the natural history and what the importance of the outcomes that we capture, and then a presentation of, at that time, we had presented through 36-week data. So Jerry Mandel provided the first presentation of our 36-week data. The late breaker session was on Saturday, and that's where we presented the 48-week data, which you'll hear Ed Kay describe today. I'd like to just briefly go over the objectives of this call, and we will hope to provide as much room and time for questions and answers. But we will start with Ed providing an update on the 48-week results that were presented at World Muscle. I will then provide the next steps for eteplirsen. This will be, again, limited to all of the things that we've disclosed previously in terms of the timing and our intention related to regulatory strategy and regulatory activity. I'll refer, I know there's a lot of interest among the community about additional clinical studies, so I'll talk about our current thinking about that I've shared on other calls and other opportunities in public forums. And then there's a lot of interest in the manufacturing and what that entails and what that requires. So we'll speak to that. And then more importantly, we know that eteplirsen only can serve a fraction of the Duchenne patients that can benefit from an exon skipping drug and the technology that Sarepta holds. And so we'll talk about our intention to develop other exon skipping targets, and we'll try to address that as well. But again, we'll try to leave as much room for Q&A. And with that, I'd like to turn over to Dr. Ed Kay. I just want to remind everybody, Ed joined us from Genzyme, which arguably established a business model in the rare disease space. So before Genzyme, many companies shied away from diseases like Duchenne because they did not believe that you could that it was worth the investment. And that was a real shame until the industry embraced the idea that we could actually have a business model where it was worthwhile to pursue drug therapies for patient populations that were low in prevalence but were very severe and where technology existed to treat them. So Ed was responsible for leading all the neuromuscular, all the rare genetic disease programs with the architect of sub-T programs like Myozyme and Alverazyme for Genzyme, and he really is intimately knowledgeable about what it takes to develop drugs and to pursue regulatory approval for drugs like this. In addition, he's a pediatric neurologist, and he had specific ties to the Duchenne community and to the broader muscular dystrophy community through his affiliations with organizations like Cure Duchenne, like the Spinal Muscular Atrophy Association, like Cure Congenital Muscular Dystrophy. So I'm very pleased to introduce Ed, who will walk through the 48-week data.
Thank you very much, Chris. So as Chris mentioned, what we're really going to do is review the data that was presented at the 2012 World Muscle Society meeting and really try to walk you through what we learned from this study. So I think the first issue that we need to just focus on is really describing what eteplirsen is, and just so that everybody understands, it is a RNA modulator and really addresses the underlying dystrophin deficiency, which is the problem that we see as far as the cause of Duchenne's muscular dystrophy. So what this drug does is it really directs alternative splicing, and specifically for deletions that are amenable to exon 51 skipping. It's administered through an intravenous infusion every week. One of the differences in this particular molecule, it is neutral. It has no charge to it, which differentiates from some other backbones. It also has a fairly short half-life, between 2 and 6 hours, and is cleared directly through the kidney. And we do have safety data based on this study for up to a year at 50 milligrams per kilogram per patient. So what we have also noticed is that the particular approach for exon skipping, as we mentioned, is really to take an out-of-frame deletion and place it into an in-frame deletion. So the out-of-frame deletion causes the protein to be very unstable. It breaks down. It can no longer be functional and can't attach to the membrane of the muscle. So by adding eteplirsen, what we're doing is we're doing an in-frame mRNA transcription and restoring the ability to produce and have a functional, a slightly smaller, but hopefully is still a functional dystrophin protein. And many people, I believe, do recall what the Study 202 design was, but just to review it, our first study was a 24-week double-blind placebo-controlled study in which all of the patients were in one of three cohorts. They were either a placebo or they were on a 30 or a 50 milligram dose. And we had alternating biopsies that were done at 12 and 24 weeks. At the end of 24 weeks, all of the patients were rolled over into either a 50 or a 30 milligram per kilogram weekly dose. So everyone knew at that time that they were on drug. However, it wasn't until after the 36-week data that patients were informed whether or not they were initially on the placebo treatment group. So really the purpose of the study was, of course, long-term safety. It was a Phase IIb study, but we also were able to address efficacy. And the primary measurement that we used was the dystrophin measurement, and this was expressed as percent dystrophin-positive fibers. And just to make clear, this is not just a change from baseline, but in fact what we did is we looked at all of the fibers and we recorded the percent positive as our baseline. We also included a functional outcome measure, the 6-minute walk test, since it has been used in the past for other muscle diseases. And then, of course, we looked at a number of safety aspects, including clinical and laboratory measures. So just to review, at the baseline, of course, as I mentioned, we had three different groups, the placebo, the 30, and the 50 milligram group. And in general, these groups were fairly well matched in regards to the age, their overall size, and also their mean 6-minute walk distance. The only slight difference was the 30 milligrams per kilogram dose was slightly older and had a slightly lower 6-minute walk test distance. So the inclusion criteria, the first criteria that we had to use is these patients had to have demonstrated a genetic defect, a deletion, that would be amenable to exon 51 skipping. Obviously, we wanted to make sure that we would only have patients who could respond potentially to our drug. We also tried to identify boys that were likely to decline during a 48-week period. So we took boys between the ages of 7 and 13 because we knew prior to that time the boys are actually continuing to improve in their 6-minute walk test distance. We also chose boys that were affected and that they had to have between 200 and 400 meters as far as their baseline 6-minute walk test distance, suggesting that they already were affected. These individuals were not asymptomatic and they had evidence of disease progressing already. We also wanted to make sure that we didn't have any confounding variables, and so we made sure that everyone was on a stable dose of corticosteroids, and they had to be on a stable dose for at least 6 months or 24 weeks prior to entry of the study. And they could not change the dose of steroids also during the study. Again, we wanted to make sure that whatever change was seen was related to our therapy and not to some other standard of care. So just so everyone understands, because there has been, I think, some confusion about the dystrophin quantification method. And again, this is expressed as dystrophin-positive fibers. So what was done is all of the boys had a surgical biopsy taken from the biceps muscle. This is both pre- and post-treatment. And there was a total of 3 biopsies that were obtained throughout the study. Also mentioned that it was done by the same surgeon, and it was the same laboratory that looked at the biopsies. A lot of fibers were counted. So there was 2 blocks, and there were 24 sections of slides. And so on average, there was more than 1,000 fibers counted per patient biopsy per visit. The antibody that we used to identify whether it was positive or not was a antibody Mandes 106, which detected the immunofluorescence. And then we had looked at the mean values of dystrophin-positive fibers. So we counted all the fibers, and then identified the positive fibers, and then divided by the total fibers throughout the slide. So we had pre-treatment mean values of dystrophin. These included the revertant fibers and also the background levels. And then we subtracted it from the post-treatment values. So all we were really measuring was the actual novel dystrophin that was produced during the study. And the results, as I mentioned before, were a percentage of normal positive controls. So it's 100% positive fibers is the control. So this is the week 48 data as was presented at World Muscle Society. And what we saw was in patients who were treated for 48 weeks at a dose of 50 milligrams per kilogram, or total, I should say, all of the boys who were treated for a total of 48 weeks had, on average, 47% dystrophin-positive fibers, which was statistically significant. This was both for the 50 and the 30 milligram doses. We didn't see any difference between the two dosing cohorts in regards to the number of positive fibers that were seen. Also, in the previous placebo-treated group that eventually went on to therapy after 24 weeks, we also saw that this group of patients had an increase in their percent dystrophin. It was 38%, which also achieved statistical significance. And if we look at the next slide, we can see the individual values for the dystrophin production seen both at 24 weeks and then through 48 weeks. So I think the people who recall the initial 24-week data, the only group that we saw dystrophin-positive fibers occurred after 24 weeks. We could not see an increase after 12 weeks, which suggested that even at the higher dose at 50 milligrams, the 3-month time period was not an adequate amount of time to produce the dystrophin. We did, however, see dystrophin after 6 months, and these values are noted on the bars. What we could see also that after a total of additional 6 months of treatment, 48 weeks, the patients who had a biopsy previously at 6 months, so you could see the dystrophin in, they continued to increase, so that we saw increased levels compared to 6 months at 12 months. Again, suggesting that it takes a longer period of time for the dystrophin production to occur. Also, on all of those boys that were changed over, both in the 30 and the 50 milligram from placebo, after 6 months, each of the patients demonstrated increased production of dystrophin, so that all of the patients in the study, after a minimum of 6 months, were able to demonstrate dystrophin. And then in addition to that, if they were treated for a longer period of time, an additional 6 months for a 1-year period, we could see increasing levels of dystrophin. And just to show the slides of the immunofluorescence, and hopefully they show up on your screens, but we did not see dystrophin present on the pre-treatment values, but at the 30 milligram per kilogram dose, the group that had been biopsied at 24 weeks, then again at 48 weeks, we were able to see dystrophin at both time periods, but there was increasing levels of dystrophin at week 48. Also, the biopsy on the samples for the cohort that had 50 milligrams per kilogram pre-treatment, we don't see any evidence of immunofluorescence, but after 48 weeks of treatment, we see the dystrophin-positive fibers. And then, in addition, if we look at the placebo-delayed treatment, we could see that for all of the 4 patients, pre-treatment and after 24 weeks of treatment, again, we could see the percent of dystrophin-positive fibers had gone up. One of the problems, I think, in trying to measure dystrophin is what is the most appropriate tool to try to measure dystrophin. And obviously there are a lot of camps who have their favorite measurement. I think what we were really interested in is making sure that we had a robust system that we could measure the dystrophin, and we could do it reliably and reproducibly throughout the study. Saying that, that's why we took the percent positive fibers of normal as our endpoint. This is after discussions with many of the world expert leaders, that was thought to be the most reliable system. However, we also looked at other methods to try to confirm that the dystrophin that we saw was indeed the novel dystrophin that we expected. So we did use the Western blot, and as you can see in this particular slide, we have evidence of patients who were on 50 milligrams, 30 milligrams, and also patients who had been on placebo and had received 30 milligrams per kilogram. And what we could see is that compared to the actin control, which is another protein in the muscle, we could see, using the Mandy's 106 staining on that top line, as you can see that all of the patients had evidence of dystrophin production on Western blot using this Western blot technology. In addition, to look at another methodology to confirm that what we were seeing on the immunofluorescence was indeed the dystrophin, we did the PCR and looked specifically for the skip product, for the product that should be the appropriate size, and we found that, again, this was an example of patients who were missing exon 49 and 50 and that were appropriately skipped. We were able to demonstrate that, in fact, on RT-PCR, we indeed did have evidence of dystrophin production, and it was the right size and quality that we expected it to be. So, in addition to the biochemical marker of dystrophin-positive fibers, we also looked at the 6-minute walk test data. And this slide goes over the entire cohort of 50 milligrams compared to the placebo-treated. And what we were able to demonstrate is that if you look at the top slide, and then this is the meters walked, and this is, and on the bottom, if we look at the x-axis, it's the time period. So it's from baseline up to week 48. So what we were able to demonstrate, and what we had spoken before, at week 36, we could see a difference between the two groups. And, of course, we were very interested, is this difference between the previous placebo-treated group and our 50 milligram per kilogram group, was it maintained? And what we demonstrated here, again, looking at that top line, is that there was no decline in the 6-minute walk test distance. And remember, we had chosen the patient population that we would have expected to decline on average of about 115 meters based on their 6-minute walk test, their age, and some of their other characteristics. So they did not lose the expected amount of distance walked during the 6-minute walk test distance. Also, importantly, if we look at the previous placebo group that now were on treatment, they did not continue their decline, they stabilized. And I think if we look at this, is that what we realized from the dystrophin data is that we did not see significant increases in dystrophin at 3 months. So somewhere between 3 and 6 months, the amount of dystrophin produced was able to be quantified, and we know that they're first have to have the dystrophin, then the expectation is a restoration of fibers and restoration of function. Then we started to see the improvement in the 6-minute walk test. The same is true in the previous placebo-treated. We did not expect to see any change because we did not expect to see any dystrophin at week 36. It was only after week 36 when the dystrophin began to be increased in the muscle biopsy, and that's when we expected to see the clinical manifestations in regards to the 6-minute walk test. So I think this was very helpful for us. The most important information is that in this 50 milligram group, we didn't see a decline over a year's time, and that also the decline that continued up till week 36 in the previous placebo-treated group stopped, and that progression did not continue. So those are really the two, I think, important aspects that we had learned from doing this particular study. Now, as we had mentioned previously, the study 201, the 24-week study, there were 2 patients who began to have a very rapid decline really within the first month of the study. So these boys, by 6 months, were no longer able to walk. They had a very rapid decline. and they could no longer be tested. So one of the problems we had in the study is that we only had 2 patients in the 30 milligram group that could continue to do the outcome measurements for 6-minute walk test. But we did continue to follow them in regards to other safety facts, and what we did is we monitored them, their pulmonary function. We also followed cardiac function in regards to echo, and these did remain stable throughout the 48 weeks. These 2 boys that deteriorated were monozygotic identical twins. They had been in the 30 milligram per kilogram group, and they were excluded from the modified intent to treat, again, because we could not do the 6-minute walk test on these boys. They did, however, show dystrophin production, and it increased between 24 and 48 weeks. And then what we saw in regards to the other efficacy measurements, we did not see any deterioration overall in these boys other than the 6-minute walk test, and we continue to follow them in regards to other safety parameters, and we'll continue to monitor their pulmonary function and their cardiac function. One aspect, of course, that we've been asked many times is there a difference between the various populations? And I think people have been concerned that perhaps only some of the boys responded to the therapy. So in order to really address this particular question, this forest plot is really to look at what happened with the various subgroups. So I think what we could see is that the largest group, of course, was the 50 milligram per kilogram, because there was 4 of these boys, and we compared them to the placebo. So if we look at the 6-minute walk test distance, it was statistically significant p-value. We also saw it was in favor of the eteplirsen treated boys, even in the 30 milligram per kilogram, although it didn't reach statistical significance because of the small numbers. And then we saw, if we look at all of the treated boys who were treated for 48 weeks, it did become statistically significant. There were a couple of groups that became interesting to look at. We did notice, however, that the boys who were younger at baseline, less than 9.5 years, and the boys that had the higher 6-minute walk test distance did the best. We still saw, however, a response to boys who were older and who had walked less at the time. So everyone did show a response in favor of eteplirsen, but we just saw an improvement. And we also looked at the 4950 genotype, and the reason for this is we had patients who were in the treatment group and also in the placebo, and given this specific genotype, we could see that there was a definite statistically significant improvement in the boys who were treated. What we did demonstrate in this, we had 5 different genotypes from the study, and we did not see any differences in response to exon skipping based on the genotype. So we do not have any evidence to suggest that there are specific deletions that respond more often than not. So to look at some of the safety monitoring that we did a lot of laboratory monitoring, and we were very interested in looking at kidney function. So one of the problems with boys who have muscular dystrophy of any form is that their serum creatinine levels go down as part of the disease because their muscle mass goes down and creatinine is excreted from the muscle. So we actually looked at both urine and serum. Cystatin C is another method to look at renal function. And these were very normal throughout the study. We also looked at kidney injury molecule 1, which looks at renal tubular function, remained normal. We did not see any evidence of proteinuria. We also looked at very specific liver function tests, the gamma-glutamyl transferase. The muscle enzymes were stable. There was no increase that we could see. Also, all of the other chemistries, such as C-reactive protein, which is a marker of inflammation, was normal. We had looked at all the coagulation profiles, all the pulmonary functions, and cardiac function by the electrocardiogram and also by the echo. All of these were normal, which was very helpful, and we had no treatment-related adverse events. If we look at all of the what we saw was we saw no treatment-related adverse events. The treatment-emergent events that we saw that occurred during the study were all thought to be related to the underlying disease. And so this was, and we saw really a similar profile for the placebo and for both of the treatment groups. So again, what we were looking for is making sure that because many of the antisense oligonucleotides have been associated with other toxicities, so we looked for the common toxicities that could be seen, and we did not see any evidence of specifically of proteinuria, changes in the immune activation, changes in complement, coagulation, drop in platelet counts, and really all of the functions remained normal through the 48 weeks. So to summarize what we found from this study is that this Phase IIb study did meet its novel endpoint, was really looking at the novel dystrophin production, which we defined as a percent of dystrophin-positive fibers. So there was a significant level of dystrophin in all of the eteplirsen-treated patients after 24 weeks. It was an average of 47% after 48 weeks of treatment and 38% after 24 weeks. We also noticed that all of the patients showed evidence of continuing dystrophin production with a longer duration of treatment. In other words, the 48-week had more dystrophin production than at 24 weeks, and this was in the 30 milligram per kilogram cohort. Looking at the 6-minute walk test distance, what we could see, there was a significant clinical benefit on the primary clinical outcome. Again, looking at the 6-minute walk test distance comparing to the placebo delayed treatment. The 50 milligram group demonstrated an 89.4 meter benefit compared to placebo for 24 weeks, followed by the 24 weeks of treatment with eteplirsen. We didn't see any statistical significant difference between patients who received 30 milligrams of eteplirsen based on the previous placebo to delayed treatment. And the treatment effect was demonstrated across key subgroups of age, baseline 6-minute walk test, and genotype in the evaluable patients, which we refer to as the modified intent to treat. So what we saw is eteplirsen also appeared to be well tolerated up to 48 weeks of treatment at 50 milligrams per kilogram. During the study, we saw no treatment interruptions or discontinuations and no laboratory evidence of toxicity. So our conclusions were that this Phase IIb study demonstrates the promise of eteplirsen to be a major advance in Duchenne's muscular dystrophy by treating the underlying cause and potentially altering the course of the disease. So this portion for the clinical review is completed, and what I'd like to do is turn it back over to Chris Garabedian.
Okay, thank you, Ed, and really appreciate that detailed overview for the group listening in. I'd like to talk about the next steps for eteplirsen and specifically our regulatory next steps. There's been a lot of interest in additional clinical studies and in what populations and when those might start, and then just questions about manufacturing and producing more drug supply. So again, I'm going to limit my comments to what we've shared previously in other public forums. We are very encouraged by these data. Going back to the 24-week data that was presented in April, the reason that companies do Phase IIb studies is to better understand how to design and power confirmatory studies moving forward. And at that time, while we had some very encouraging data in the production of dystrophin in the 30 milligram cohort that we did biopsies at 24 weeks, we did not have a statistical benefit on 6-minute walk, and we didn't have enough information on what we felt would be the true assessment of a treatment effect and how to best design and power a study moving forward. So at that moment, we decided to wait and see how Study 202, the extension study, would play out and what type of treatment benefit we would continue to see. And we knew that we'd have an opportunity at the 48-week time point that Ed described of understanding if we see consistency and robust levels of dystrophin across the entire patient population, including those placebo patients who rolled over. So as we've discovered this, what we believe is a very strong data set, we have started to think about the broader program and what might be required from regulators to get the drug approved. And we also believe that the results that Ed has highlighted are unprecedented in terms of the consistency of the dystrophin produced across the sample population, as well as the benefit on 6-minute walk, and the fact that we achieve statistical power and a p-value on both the primary endpoint of dystrophin-positive fibers as well as the primary clinical outcome measure on 6-minute walk, gives us an opportunity to have a real dialogue with the FDA about faster paths toward approval. We know this is a progressive disease. We know there are patients that believe they would benefit from this drug right now. However, we need to talk to the FDA to understand how they feel about this data set and what they believe is required and necessary to figure out the fastest path to approval of this drug. So right now, we are still continuing to analyze the 48-week data set beyond the data that we were able to prepare and share at World Muscle and what Ed shared today. We are preparing briefing documents of this data set, and we are contemplating the right questions and objective of the meeting request with the FDA. That's an important part of the equation, too, is to make sure we set the appropriate context for the next meeting with the FDA. And we want to ensure that we have the sufficient data to support the objective that we have going into the FDA. So as I've communicated previously, we are preparing those data summaries and those briefing documents. We were preparing for a request for a meeting with the FDA that we hope will occur around year-end. I've also communicated that it is likely very difficult to calendar something by the end of the year, and more likely it will be calendared early next year, in January or February of next year. So we are, again, continuing to prepare for that. We will not have more communication or direction about the pivotal trial or confirmatory trial, additional data that would be required to get the drug approved, or any additional commitments as part of this program until we're able to have that discussion and meeting with the FDA. So at this time, while we know there's a lot of interest and a lot of demand for eteplirsen today, we are preparing all of our activities and every ounce of our effort to figure out how we can be on the fastest path toward approval of eteplirsen, so we can get this drug in the hands of every patient that can benefit. Every age group, every status, whether ambulant or non-ambulant, and we really need to talk to the FDA about how to achieve that in the most expeditious way possible. So the additional clinical studies that we've highlighted here will be determined after we meet with the FDA and understand what they're going to require. This goes hand in hand with our manufacturing strategy. So as I've communicated, going back to April after our 24-week data, is that we had to put off our scale-up plans until we knew that this was a drug that could be pursued into pivotal studies, because the small number of patients we had showing dystrophin at 24 weeks wasn't enough information to give us the confidence to pursue this very aggressively in a confirmatory or pivotal study. When we had the 36-week data, we started, that was announced in July, we started efforts to prepare for scale-up of what we describe as mid-scale production to allow us to support a confirmatory study that would be initiated in the second half of 2013, and that means enrolling would start in the second half of 2013, with dosing to begin in early 2014. And that was based on mid-scale production, of which, again, is underway in terms of the plans. This is a long process. It's costly. It's timely. There is setup required. There is validation of the methods that we use to scale up. We have to work with contract manufacturers to get to that level of scale. And then we have to have validated batches, and typically the FDA has required 3 validated batches using GMP methods and processes. And then we have to put those drug vials on stability and do stability testing before it's released to be dosed in patients. And so this is a huge effort. It is going to span throughout 2013 so that we can have drug ready to dose patients in a confirmatory or pivotal study beginning in early 2014. Now, in parallel to those efforts for mid-scale production, we are also thinking about commercial demand. And that would be, how do we satisfy demand that would come if we were to get an early approval or an accelerated approval by the FDA? Now, we are in the planning processes right now for what would that look like and what would that take. And until we have that meeting with the FDA to understand the feasibility of getting this drug approved early, before we had confirmatory evidence in an additional clinical study, we will be prepared. And if it looks promising, then we will begin in earnest the efforts to scale up to what we describe as large-scale production. We will not know until we have that meeting with the FDA the timeline related to having drug ready. Again, we would have to submit large-scale production, validated methods, the stability data, et cetera, in order to use drug from those large-scale productions. And so that could be happening in parallel. But again, we are right now just focused on mid-scale production, and that would net product to be used for additional clinical studies in early 2014. That is the guidance we've provided to date. And again, we will provide additional information as we get it through other forums, publicly announced earnings calls, conference calls, and investment conferences. With that, I'd like to just move to the question about additional exons. And we have already begun efforts to pursue additional exon targets that we previously announced on exon 45 and exon 50. We are in the throes of those activities, and we hope to accomplish the pre-IND work required to submit an IND by the end of next year. And so, again, we've already begun those activities. Those activities will continue throughout 2013 with a goal of having an IND filed for those 2 additional drug targets by the end of next year. We have also been in discussions to pursue other exon targets. We have been in discussions with granting authorities in the EU on exon 53, and we're hopeful that we may receive grant money at some point to support exon 53 IND-enabling work. And then there has been foundation interest in helping us with exon 44 and potentially additional support for exon 53. And again, we've been engaged in discussions to get support for that exon. The goal is first and foremost to pursue eteplirsen approval. And following that will be a strategy that pursues these additional, 4 exons that comprise the top 5 exons. If we are able to work with regulators with the proof of concept where we can show that there is safety regardless of the sequence that we're targeting, that there is the same pharmacokinetics regardless of the sequence that we're targeting, that we establish a standard dose across every exon target, regardless of the sequence that's attached, and we're able to show dystrophin production and clinical outcomes across these top 5 exons, then that is something we would like to discuss with regulators, how we would pursue rare exons, those that are infeasible to do the safety and efficacy studies that would support claims and powered sufficiently, and how we could get those drugs approved to enable what we call a class approval, where we could see the ability to produce all the exon targets that are needed to make sure every DMD patient that could benefit from our technology could actually have an opportunity to take drugs that could improve or halt the course of the disease. So with that, Sharon, I'd like to open it up to questions.
Oh, thank you, Chris and Ed, for that presentation. So I just want to remind everyone that we solicited questions before the webinar over a period of, from the time it was announced, and we received quite a few from all of you. And these have been sort of distilled into key questions that we'll go over now. If you want to write a question into the chat box, you're welcome to do that. We will capture those questions, and if they are not answered in this Q&A session, we'll try to get answers to them later. But we're going to start by addressing these questions that during the webinar that were submitted in advance. So just to start, of course, muscle strength improvement is important, but people are probably wondering if eteplirsen has shown any benefit on other measures like pulmonary or cardiac, which are very important.
Thank you, Sharon. Obviously, we certainly agree. We know that Duchenne's muscular dystrophy affects the heart, and certainly it affects the pulmonary system because of the intercostals and also the diaphragm. The problem that we experienced in the population that we were studying, we were primarily focused on the 6-minute walk test, and all of the boys at the beginning had normal pulmonary function and had normal echocardiograms and ECGs. So we did not expect them to deteriorate during this time. What we can say is that, as you probably know, the 6-minute walk test is not only for muscle strength, but it does measure endurance. So the expectation is that the pulmonary and cardiac systems would have to remain normal in order to keep a baseline 6-minute walk test without deterioration. The only thing we can say is during the period of study, there was no evidence of deterioration in pulmonary or cardiac function. However, they were all normal to begin with, and so I think it's too premature to say what will happen long term. But this is information that we will continue to collect on the boys, and hopefully as we go farther out, we'll have more and more information to say about lung and cardiac function.
Thank you. So from the time the boys start taking the drug, there's a lag before you see dystrophin made. Why do you think it takes so long to make dystrophin?
Yeah, that's actually a great question, Sharon. There's a couple of reasons, and of course we can't say we know 100%, but we do know a few things about dystrophin. We know it's a very large protein. It's the largest protein in the body. And then it has a fairly long half-life. So it has a half-life of about 2 or 3 weeks. And we know from drug studies in the past that in general it takes about 5 half-lives to reach a steady state. So that's one reason. It takes a while for the drug to be made and to reach a steady state, so there's certainly a delay. The other aspect, of course, is that we know that there is initially, based on the animal work, some patchy distribution of the drug when it gets to the muscle. Over time, there's a greater and greater distribution of the drug in the muscle, and as we've seen in this study, there's an increased level of dystrophin production. So I think what happens is the dystrophin is probably there. We know that low levels of dystrophin has been seen in other studies are very hard to quantitate. But then over time, and somewhere after 3 months and before 6 months, we see an increasing level of dystrophin that we can actually quantify and measure. And then once we see that dystrophin there, presumably we would expect to see the clinical benefit.
And then speaking of the amount of dystrophin, do you have a feel now for how much dystrophin is needed to produce a change in the 6-minute time walk? That obviously is a challenging question, and I think what we have based on a collection of evidence from previous studies, this includes animal studies, it also includes studies in boys with Becker muscular dystrophy, the milder form of muscular dystrophy in which there is some dystrophin. There seems to be a consensus that about 10% is a threshold
in which you start to see a clinical effect. And we know that in some of the animal studies, even levels as low as 3 to 5% may show an improvement in treadmill distance, for instance, for mice, for animals that were treated. But I think what we expect is that there probably is a minimal amount of dystrophin that you have to have. You don't need 100% to have a response in regards to your improvement in muscle function. We also know that, just as I think everyone has mentioned, it's an imperfect method. There's a lot of sampling variability that occurs. So there is no one dystrophin level that correlates with a 6-minute walk test. It's not that linear of a process, and it just doesn't predict a 6-minute walk test distance. I think what we know is you have to have some dystrophin. We think the best estimate is you probably have to have over 10% to see an effect. And how much more you get is still, I think we don't know. But once you reach that threshold, then it appears that you're starting to see a clinical benefit.
So do you think if eteplirsen is approved that steroid treatment will still be recommended and continue to be used alongside eteplirsen?
That is a question that we've been certainly asked in the past, and unfortunately, for this particular study, we really can't answer that. I mean, I know that people are very concerned about the side effects of steroids and the litany of side effects, the hypertension, the weight gain, the changes in behavior, and all of these things that people would love to avoid. But remember, our study was done on boys who were on a stable dose of steroids, and so we can't really answer that question. Obviously, everybody would love to be able not to have to use steroids and have an alternative, but I'd say for right now, we really can't answer that question.
Well, another question along those lines, if eteplirsen were approved, what do you think the recommended age would be to start the drug?
The FDA, of course, will typically help you shape the label in all, certainly in the United States, but really throughout all countries. The physicians are guided by what's in the label. I think what initially, we would expect that it would be guided by what the data showed. And so what we can say based on this study is that boys who were younger and who had a greater 6-minute walk test baseline distance did seem to respond. So I think, logically thinking, the expectation would be that boys who were younger and were healthier might do better. But I think that's a discussion we would have to have with the FDA to see what the label would look like. And also it would be up to the individual physicians to make that decision based on the results and looking at the individual patient.
Okay, so here's a question about side effects, which you may have partially addressed in your presentation earlier. But what side effects have you seen in the study, and do you have any thoughts on long-term safety? And do you have studies planned to evaluate long-term safety?
We have, and we will be actually having a presentation at the Oligonucleotide Therapeutics Society. We are looking at long-term safety, 9-month safety in animals, and looking at primates. But also we do have, obviously, 48-week data, and based on this study, we have not seen any kind of signal that points to any particular organ system that appears to be affected by this drug. So right now, we haven't seen anything. We will obviously continue to monitor these boys and to monitor the boys who are non-ambulatory and continue to collect safety data, hopefully for years, to be able to understand. We do have, we have now 38 patients who've been treated with eteplirsen for Duchenne's muscular dystrophy, but we have over 400 patients who have been treated with PMOs for other indications and other studies, and we have not seen any specific drug-related adverse events. So we'll continue to look, but right now we haven't seen any signals that suggest there are any particular toxicities, and specifically we haven't seen the toxicities that have been fairly generic for some of the other backbones.
Okay, so a question about dosing. What dose are you planning to pursue for additional studies or for approval, the 30 mg or the 50 mg per kg? And is it clear that one dose is more effective than the other?
Well, I think obviously we realize this is a small study, and there were only 4 boys in each of the treatment groups. Based on our primary outcome measure, which was the percent of dystrophin-positive fibers, we did not see a difference between the 30 and the 50 milligram dose. And in fact, the 30 milligram did a little better than the 50 milligram after 48 weeks. And also, if we look at patients who were on placebo and were rolled over onto 30 or 50, we also couldn't see a difference in the dystrophin production. So based on the dystrophin production, we really didn't see anything that would suggest that one dose was better than another or that we would have to use a higher dose. I think the 6-minute walk test distance, which of course everyone has focused on, is we did see a better 6-minute walk test distance in the boys who were treated with the 50 milligram group. But remember, unfortunately, 2 of the boys had the rapid deterioration shortly after beginning the study, so we weren't able to collect 6-minute walk test distance long term on that. So we really can only compare 2 boys who were treated with 30 compared to the boys who were treated for longer, the 4 boys who were treated for 50 milligrams for a total of 48 weeks. What we can say is that if we look at all of the treated boys, as we showed in the subgroup analysis, they all did better than placebo. So I think at this point, we really don't have any evidence to suggest that one dose is better than the other, and it's looking as 30 milligrams is as good as the 50.
Okay, so here's the question that everyone wants to know. Are you planning to ask the FDA for accelerated approval? And when will you let the community know if the FDA agrees to that approach?
Yeah, Sharon, this is Chris. I'll answer that one. So we know that there's a lot of interest around Sarepta's intention to pursue an accelerated approval filing. What we've tried to say to this point is that we are well aware of what the FDA's authority is related to approving drugs under accelerated approval guidelines, and we know that there's been a lot of broader interest, even from Congress, in their recent passing of the FDASIA Act, the PDUFA 5 legislation, where there was really expressed language around encouraging the FDA, in so many words, of using this authority that they have, particularly in the areas of rare disease, where there is no approved treatment and no disease-modifying ways to stop or halt the progression of a disease like Duchenne. So we're very encouraged by our data set that it may be something that the FDA would accept under an accelerated approval filing, but it's in our best interest to really maintain good dialogue with the FDA, good relations with the FDA, and that's something that we need to work with the FDA together on to find the fastest path toward approval. We don't want to be presumptuous, and we really need to get their guidance and their feedback of whether that's feasible. In terms of when we would know, when we calendar a meeting with the FDA, we will look for an opportunity to, alert the community of when that date would occur. But also, oftentimes, the type of feedback you get at the FDA meetings have room for interpretation. And what we want to ensure is that we have full agreement, and these are usually in the form of meeting minutes, right, that, we would provide, they would provide to us. We talk about those to make sure there's a meeting of the mind and that both parties are in agreement of what was discussed, what the answer to those questions actually are. And those are, established in the form of formal minutes, proprietary minutes between sponsor and FDA. And once we have a clear understanding and a meeting of the mind, then we will communicate if there is an intention to pursue an accelerated approval NDA we would know likely within about 30 days from that meeting that would be calendared. So, look, we understand the interest and the anticipation of this knowledge, and we are too. We share the community's interest in this, and as soon as we have an intention to do so, we will communicate that.
So a related question is, if you do future clinical studies with eteplirsen, can you describe what those might look like?
Yeah, as I mentioned earlier, we really need to get FDA feedback, and we want to make sure that any drug that we dose in patients is part of a broader program that would be supportive of a FDA approval for this drug, so we can ideally get the drug on the market so that as many patients as possible can benefit from it. And so while we have a lot of information that we've gathered from the current clinical program that will help guide our discussion with the FDA and guide our thinking around confirmatory studies, we don't have any guidance at this time for what those studies would look like. But of course, we want to make sure that we achieve as broad a label as possible, because we believe that if a benefit is shown, in this case, in the Phase IIb, we showed it in enrolling patients with inclusion criteria that included 7 to 13-year-olds who were ambulant and amenable to exon 51 skip. Obviously, we know that starting the drug earlier may provide even better benefit or chance to slow or halt the progression of the disease and alter the course of the disease potentially. We know that patients who are older than 13 or who are non-ambulant, it's still a progressive disease that's based on the lack of dystrophin. That's not lost on us. We hope that's not lost on the FDA, that just because we're hitting patients at the time that they would lose ambulation doesn't mean that the restoration of dystrophin won't help those patients. And we're fortunate in that we're capturing some non-ambulatory information. In the 2 boys that showed rapid progression, they're in a non-ambulant state now. We're collecting good safety information, good dystrophin information, good outcomes, upper arm extremity and other outcomes like pulmonary and cardiac function in these boys. So we think that that's very informative to go into the FDA to help them understand what the drug utility might be in the broader patient population. But at this time, we don't have any specific guidance to provide on what those clinical studies will look like and when they would start.
So along the lines of non-ambulatory patients, will they be able to get access to eteplirsen?
Again, we will need to talk to the FDA to understand what is required to get this drug into all of the DMD patients that could benefit, and that includes the non-ambulatory population, okay? We believe this is the same patient, okay? They're amenable to exon 51. They have a progressive disease that's marked by the lack of dystrophin, and we believe they will benefit on restoration of dystrophin. And so we need to have that dialogue with the FDA to understand what requirements they will have to achieve the type of broad label that would allow us to get the drug approved and be able to be used in non-ambulatory patients as well. So, that's our intention. It will be part of the discussion of achieving that type of broad label when we meet with the FDA. And again, we don't have any more information to provide at this time.
So how much do you think this drug will cost?
Sharon, first of all, the cost of the drug, okay, which is what it costs to produce and make the drug and put it into vials so that they're on the shelf, at the clinic or pharmacy, is something that will hopefully be improved as we scale up production. So the drug is very costly at the small scale that we're producing it at currently. We expect that mid-scale production that would be able to provide the drug for additional clinical trials would be a reduced cost than what we're currently paying to produce the drug. And then we hope to achieve even more cost efficiency upon a commercial scale when we have large-scale production batches. But no matter what, this is a large dose burden, okay, 30 to 50 mg per kg weekly in patients. It is a novel drug production process. There's been no experience in scaling up morpholinochemistry as an oligo. This is a synthetic peptide-like backbone. So it's differentiated from other oligos like mifemersen, for example, that are going through scale-up currently for the launch of that drug. And so, we're doing this for the first time, and it is costly when you do anything for the first time to make sure you have the processes validated, that you're doing things in a stepwise fashion, and that you are ensuring that you have good stability, good purity profile, and consistency batch to batch when you start to scale up production. So we do not disclose the specific cost of eteplirsen, but it is an expensive drug. And the only reason that companies can pursue technologies like this that have cost of goods that are very high is when you're pursuing them for a rare disease like Duchenne, where there is a business model that supports it. So, that's the cost of eteplirsen question. If you're asking about the price of eteplirsen, I think you can look to other low prevalent diseases, and the price point there is well known in terms of companies like Genzyme or Shire or BioMarin or Alexion more recently, or Viropharma, and or Vertex with their cystic fibrosis drug. Again, those are very they're highly priced drugs, but they're able to receive reimbursement because of the low prevalence of those disease areas. So again, it's too early and premature to figure out what the cost of eteplirsen is at this point, or what the potential price point is. But we also expect that the value that we're providing to a particular disease in terms of serving the unmet medical need will also be a factor in determining the price of the drug.
So I see that we're a little bit over the hour now. We have about 4 remaining questions, and I think we'd like to try to get those answered, and those, on the line who can stick with us are welcome to do that.
Sure.
So what is your plan to address the other deletions with your exon skipping technology? Which ones will go first, and what is the sequence specifically? And how will you address the really rare exons? I think you touched on this earlier.
Yeah, so Sharon, we did. I did touch on it earlier, and basically the top 5 that we shared in the slide are the ones we're focusing on to prepare for clinical development. And again, we have the collaboration with Exon 45 that was based on a couple grants that the Children's National Medical Center in D.C. and Carolinas Medical Center in Charlotte both had DoD and NIH grants respectively to develop an Exon 45 target. That's how we chose that exon. We applied for a trend grant to pursue exon 50, and we had done some optimization work on the sequence for 50 that we believe would be attractive to the NIH, that we were further along in development of exon 50 is why we pursued exon 50 in that grant, and we did receive that grant, so we're very encouraged by that. There is an EU grant that has been earmarked for exon 53, of which we, again, are hopeful that we could be the sponsor to pursue the IND-enabling work and early clinical experience with exon 53. And as I mentioned, we have foundation interest in exon 44 and some additional help with exon 53 that we're in dialogue around. So those top 5 are our priorities. They are more prevalent exons to allow us to enroll patients in clinical studies. And then we believe that if we have the proof of concept and the consistency across eteplirsen and these other drug candidates, that we could then talk to regulators about the fastest path towards rare exon approvals. Our goal is to have our drug technology available for every patient, even the rare exon, who can benefit from exon skipping technology. And we are pursuing that as aggressively as we can in the best way we know how, and we believe what we've outlined is the fastest path to get this technology approved for the broadest population possible.
So let's talk about other types of mutations other than deletions, specifically thinking of duplications here. Are these addressable with your technology?
We know that there's some academic researchers who are doing some work on duplications who believe that there is applicability of splice switching technology to address duplications. We believe that this is in the earlier stages of feasibility and that we would need to really explore that further. We know and believe that exon skipping for mutations that can be treated with a single exon skipping drug, we believe that's reproducible, and that's why we focus and prioritize that. But we are engaged with academic collaborators who have done some experiments and are interested in doing some additional experiments on seeing if this technology could be applied to duplications. And again, stay tuned for any additional information as we get it in terms of the feasibility of that moving forward.
Okay, and then can you talk about your plan to include Europe in further clinical studies or in terms of seeking approval for eteplirsen?
Yeah, as we've communicated previously, we had a European patent opposition hearing last November in which we opposed a patent out of the University of Leiden that was licensed for to GSK. We were very encouraged by the outcome of that in that we believe we enhanced our freedom to operate in Europe across the broader DMD program and most of the exons that were named in claims in that patent. However, we did not prevail on two exons. Exon 46 was one of those of which many of those patients can be treated with an exon 44 drug, and so that's why we have exon 44 as one of the top exons we're pursuing. But unfortunately, the other exon we did not prevail on was our lead program, eteplirsen, exon 51. So again, we are interested in exploring ways either through appeal or through cross-license in terms of how we might get right the ability to develop and commercialize eteplirsen in Europe. But again, at this time, we would likely be met with an infringement claim from our competitors if we tried to pursue that approval in Europe. So that's where we stand in Europe. But again, we intend to pursue all of the other exons that I've named as global development, including in the European market.
Okay, another important question. Would Sarepta consider providing eteplirsen through a compassionate use program?
Yeah, this brings us back to what I highlighted earlier, which is the manufacturing capacity. And we have focused, this is the first time, again, this company or anybody in the industry, in the world, has scaled up this type of morpholinochemistry, for clinical trials, let alone commercial or compassionate use applications. And so the drug supply is very precious in terms of how we use that, and we need every vial that we produce to be used to support the registrational program and the clinical studies required as we discuss those with the FDA to get the drug approved for the broadest population possible. The more we are successful in getting this drug approved, the easier it will be to even expand and broaden the manufacturing capacity because if we can get a commercial approval of which we can generate sale of the drug, then that gives us a lot more flexibility to, again, consider these broader populations for the drug. Then that goes beyond eteplirsen. So at this point, we have to figure out what the requirements are with the FDA, how much drug supply we'll be able to produce, and by when, with the mid-scale and plans for the large-scale production. And depending on what those requirements are with the FDA, what additional drug supply, if any, we would have, then we would look to figure out how can other patients benefit from this drug, beyond the clinical program.
Okay, finally, last question. You're talking to a very supportive audience right now. What can parents do to help at this time to ensure that patients who could benefit from eteplirsen get access to the drug as soon as possible?
Yeah, well, first of all, I would say we as a biotechnology company, we know what we do well, and that is drug development and the understanding what's required in working with regulators and the FDA to get drugs approved. That is our primary focus. We have the same sense of urgency as the community in getting this drug approved in the most expeditious way possible. And it's our job to make sure we're doing that as best we can, and that we work with the FDA and hopefully provide them the flexibility, and that they will provide the flexibility to work with us as partners to get this drug approved for as many patients that could benefit as soon as possible. So that is our focus right now. We know that the community has a strong and important voice, and we know that they would like to see this drug approved very quickly. So we, again, understand their perspective, and we understand their willingness to do whatever it takes to help us achieve that end of getting this drug accessible to as many patients that will benefit. We know that and believe that the patient voice is something that has really been in the background in our industry. And we do believe that they have an important voice, and their voice should be heard. They're dealing with these diseases on a day-to-day basis. They understand the risk-benefit. I have every parent that I've spoken to, patients that I've spoken to directly, every advocacy organization I've spoken to. They're very astute, they're very engaged, and they're very knowledgeable, okay? In many times, they're as knowledgeable about the science and the trade-offs as the clinicians that I meet with or the industry partners that I meet with, because they read the literature and they understand these technologies. They understand the safety risks. They understand the challenges with development. And I do believe they deserve a voice at the table with regulators, because I think, and the FDA, to their credit, right, has opened their doors to say, we want to hear from patients. We believe they need a voice and have a voice at the table when we consider drug approvals for these very severe unmet medical needs, in which parents may be out there saying, we understand the risk-benefit, and we think the benefit of getting access to this drug outweighs the risk of what we may not know about it and may achieve with extended knowledge and broader patient populations, et cetera, moving forward. So what we understand that, but we're focused on our job, and I would tell the parents and the patients out there who want to help that there are great organizations out there, many of them, who are focused on making sure that the voice of the community is heard at the FDA. There's organizations like PPMD that are offering this forum for us to communicate to the community that, again, they have a sophisticated approach. Many other organizations are helping with these efforts, and I would tell every parent and patient, if they want to help, go to those organizations, ask them how you can help, and an organized, coordinated effort is the best way to have the biggest impact in our mind. And we continue to talk with parents and advocacy organizations. We want that feedback loop. We want to hear what they're thinking so that they can help us make sure we do drug development the right way, that we do the right trials, and that we take into account the patient's voice and the parent's voice in how we conduct ourselves as drug developers. And that's what I would say at this time, Sharon. And we look forward to future opportunities to work with PPMD and engage the community. And we really deeply appreciate and thank PPMD for the opportunity to have this dialogue. On behalf of Sarepta, Ed Kay, myself, and the rest of the Sarepta team, we really are grateful for the opportunity.
Well, thank you so much, Chris and Ed, for this update. We really appreciate the fact that Sarepta does take the time to update the community, and we know that, that's not the policy for every company and every drug development program out there in the world, that this is a sort of unique and unusual opportunity, and we really appreciate it. we also appreciate the opportunity to support, along with the Muscular Dystrophy Association, travel for participants in this study as well. I'd like to say Pat had to leave, but I'm sure she would also join in with me in giving you her personal thanks for really dedicating everything you have to this drug development program. I want to quickly remind everyone that we will capture the questions that have come in on the chat. We will see what we can do about getting those answered in some other format. So thank you very much, Chris and Ed. We really appreciate it.
Thank you, Sharon.
All right, thank you. This will end our presentation now.